Let's talk about where T cells go to look for infections, what type of regions of the body they patrol, and what they recognize, what T cells identify. Let's first go back and talk about B cells. You remember B cells with their B cell receptors. Right? And B cells, naive B cells, will go into the lymph nodes and look for pathogens, right? Viruses, bacterium, anything that's drained into the lymph node, and they'll look at any molecule on their surface. Carbohydrates, proteins, lipid, any shape that can lock into the antigen binding sites of the B cell, that B cell can bind, activate, release antibodies, cover that thing, neutralize it, opsonization, activate complement, all sorts of great things. Right? So when we talk about B cells activating, and recognizing pathogens, they're recognizing pathogens that are present in your extracellular spaces, right? Your fluid, your lymph, your humors, in your blood, in your mucus, right? That's where antibodies really do their job, and that's where B cells look for things. So that's humoral immunity is devoted to patrolling your extracellular spaces. With T cells, it's a little different, right? With T cells, we know that T cells use their T cell receptors to look at not carbohydrates, not lipids, not even whole proteins, little tiny fragments of proteins we're going to call peptides that range anywhere from 8 to 25 amino acids long. Right? And these peptides, well, where do they come from? That's a good question. So they come from inside the cell, but there are two places that inside the cell they can come from. Two different places that represent two different regions of the body that the T cells basically patrol, if you will. One part is the cytoplasm of the cell. So in the cytoplasm, you can have a pathogen, right? Usually like a virus, right? Viruses all have to get inside the cell. In the cytoplasm, they usually release their genetic material, either DNA or RNA. The viral genes are turned on, the viral genome is replicated, the virus makes some proteins, and the virus can package itself and produce some progeny. Right? So, if T cells want to know if anything foreign is in your cytoplasm, they will look on peptides that are shown on something called the MHC class 1 complex, or the class 1 molecule. It means the same thing. Right? These MHC class 1s are presented to the CD8 positive T cell. And the way we can see that here is that the CD8, when we talk about CD8 positive, CD8 is actually a protein that's present on the surface of a T cell. And it participates in an interaction with the MHC1 protein. So you can see here, CD8 is actually interacting with binding the MHC1. So it's grabbing the cell and it's making the MHC1 show its peptide to the T cell receptor. So when we talk about CD8 positive T cells, they're really looking at intracellular infections, right? And inside the cell is, when we talk about intracellular, we really mean the cytoplasm, right? What lives in the cytoplasm? Viruses, right? The other type of T cell, called the CD4 positive, or the helper T cell, well, it has a protein on its surface called CD4, right? And that CD4 protein is going to interact with the MHC class 2 complex. And this is going to allow CD4 positive helper T cells to look at peptides that came from the extracellular spaces. Well, what comes from the extracellular spaces? Well, sure, it could be viruses. But these are not things that are in the cytoplasm of the cell. These are pathogens that are taken in via phagocytosis or receptor-mediated endocytosis. So, for example, if a cell, like a dendritic cell or a macrophage, phagocytoses a complement-covered or antibody-covered pathogen, like a virus or a bacterium, right? It'll take that pathogen into the vesicles, into the vesicular system of the cell, where the pathogen is broken down. The viral or the bacterial proteins can be broken down into peptides, and those peptides are shown to the CD4 positive helper T cells. So really, when we talk about where do CD4 cells really look for their pathogens, pathogens, the source of those pathogens are from the extracellular spaces. And those pathogens are brought into the vesicles, into these antigen-presenting cells, and then the peptides are shown to CD4 positive helper T cells. So there's a difference between intracellular infections and extracellular spaces. CD8 positive cells look at the intracellular infections, typically viruses that are living in the cytoplasm. Can CD4 cells also look at viruses? Sure, but it's the viruses that are captured via receptor-mediated endocytosis or phagocytosis. So the viruses technically aren't inside the cytoplasm of the cell. They're inside the vesicular system of the cell. So CD8 cells are going to be looking for viral infections, whereas CD4 positive cells are going to be looking at pathogens taken in from the extracellular space by phagocytosis or receptor-mediated endocytosis. That could be viruses or bacteria. This will require us to understand the process of antigen processing and antigen presentation. So there'll be a whole nother video on, actually a number of videos, on how antigens are processed and presented on MHC class 1, and then a separate video for how antigens are processed and presented on MHC class 2.